Genes hold the blueprint for life. They dictate the characteristics of every organism, from the color of a flower to the height of a human. But genes themselves are just instructions. To carry out these instructions, cells engage in a complex molecular dance called protein synthesis. Protein synthesis is the process of creating proteins, the workhorses of our cells, using the genetic code embedded within DNA. Transcription is the first crucial step in this intricate process. It is the act of transcribing, of copying, the genetic information from DNA into a messenger molecule called RNA. Imagine DNA as a master recipe book locked away in a vault. Transcription is like copying a single recipe onto a note card, allowing it to be carried out of the vault and into the bustling kitchen of the cell. This transcribed RNA molecule, called messenger RNA or mRNA, carries the genetic message from the DNA in the nucleus to the ribosomes in the cytoplasm, the protein synthesis machinery of the cell. The ribosomes then read the mRNA and use this information to assemble amino acids into proteins. Thus, transcription acts as a critical intermediary, bridging the gap between the genetic code in DNA and the production of proteins that carry out cellular functions. The initiation of transcription is a tightly controlled process, ensuring that only the necessary genes are transcribed at any given time. This process begins with the arrival of RNA polymerase, a molecular machine that unwinds the DNA double helix, exposing the sequence of bases that encode the gene. This enzyme, RNA polymerase, is the key player in transcription, responsible for reading the DNA sequence and synthesizing the corresponding RNA molecule. Specific sequences in the DNA, known as promoter regions, act as landing sites for RNA polymerase, signaling the start of a gene. These promoter regions are like signposts, indicating to the RNA polymerase where to begin transcribing. Once RNA polymerase binds to the promoter, it begins to unwind the DNA helix, creating a transcription bubble, exposing the template strand of DNA. Within this bubble, RNA polymerase begins to build a complementary RNA strand, selecting RNA nucleotides based on the DNA sequence. Each DNA base, adenine A, thymine T, guanine G, and cytosine C, is paired with its complementary RNA base, uracil, U for adenine, adenine for thymine, cytosine for guanine, and guanine for cytosine. This ensures that the RNA molecule carries a faithful copy of the genetic code from the DNA. Once the RNA polymerase has initiated transcription, it moves along the DNA template strand, continuing to unwind the helix and adding RNA nucleotides to the growing RNA chain. This process, called elongation, is a meticulously choreographed dance between the RNA polymerase and the DNA molecule. The RNA polymerase reads the DNA sequence, one base at a time, and selects the corresponding RNA nucleotide, adding it to the 3' end of the growing RNA molecule. As the RNA polymerase progresses along the DNA, the newly synthesized RNA molecule begins to peel away from the DNA template, allowing the DNA helix to rewind behind the enzyme. This continuous process of unwinding, reading, and synthesizing ensures the efficient and accurate transfer of genetic information from DNA to RNA. The RNA molecule grows longer with each step, carrying with it the genetic instructions encoded in the DNA sequence. Section 4. Termination, the end of the line. Just as there are signals for the start of a gene, there are also specific sequences in the DNA that signal the end of the transcription process. These termination sequences act as stop signs, instructing the RNA polymerase to cease transcription and release the newly synthesized RNA molecule. When the RNA polymerase encounters a termination sequence, it detaches from the DNA template, releasing the completed RNA transcript. The RNA polymerase is then free to locate another promoter region and initiate the transcription of a different gene. The termination process ensures that only the necessary portion of the DNA is transcribed, preventing the production of overly long or aberrant RNA molecules. The newly synthesized RNA molecule, now carrying a complete copy of the genetic instructions from the DNA, is ready to move on to the next stage of protein synthesis, translation. This process is where the genetic code carried by the mRNA is used to assemble amino acids into proteins. Section 5. Fine-tuning, the message post-transcriptional modifications. Before the messenger RNA or mRNA can exit the nucleus and engage in protein synthesis, it undergoes a series of essential modifications, often referred to as post-transcriptional modifications. These modifications serve to protect the mRNA molecule, enhance its stability, and ensure its efficient translation into protein. One such modification is the addition of a protective cap to the 5' end of the mRNA molecule. This cap, a modified guanine nucleotide, helps to prevent the degradation of the mRNA by cellular enzymes and assists in the binding of the mRNA to the ribosome during translation. Another crucial modification is the addition of a poly-A tail to the 3' end of the mRNA molecule. This tail, a string of adenine nucleotides, further protects the mRNA from degradation and plays a role in the export of the mRNA from the nucleus to the cytoplasm. In addition to capping and tailing, mRNA molecules also undergo splicing, a process in which non-coding regions, called introns, are removed from the mRNA sequence, leaving only the protein coding regions, called exons. These modifications ensure that the mRNA molecule is properly processed and prepared for its crucial role in protein synthesis. Section 6. The power of transcription, implications for health and disease. Understanding transcription, the intricate process of copying genetic information from DNA to RNA, is paramount for comprehending the complexities of gene regulation and its implications for health and disease. Transcription is the first step in the flow of genetic information, a crucial link between our genes and our traits. Dysregulation of transcription, where genes are either overexpressed or silenced inappropriately, can disrupt cellular function and contribute to the development of various diseases, including cancer, metabolic disorders, and neurodegenerative diseases. Cancer, for instance, can arise from mutations in genes that regulate transcription, leading to uncontrolled cell growth and proliferation. Conversely, a deeper understanding of transcription can pave the way for novel therapeutic interventions. By targeting specific transcription factors or manipulating the transcription process itself, researchers aim to develop new treatments for a wide range of diseases. Gene therapy, for example, holds the promise of correcting faulty genes by delivering functional copies into cells, potentially revolutionizing the treatment of genetic disorders. Therefore, unlocking the secrets of transcription holds immense potential for both understanding the fundamental mechanisms of life and developing innovative strategies for diagnosing, treating, and preventing diseases. By unraveling the intricacies of this essential process, we gain a deeper appreciation for the power and elegance of the molecular machinery that drives life.